This video is your introduction into DNA. We're going to start pretty basic and work our way up to a more complex understanding of DNA. So let's start from the beginning. What does DNA stand for?
DNA stands for deoxyribonucleic acid. Who discovered DNA? James Watson and Francis Crick are
The two people who you most often hear.
but contributions were also made by Rosalind Franklin and Maurice Wilkins.
So what is DNA made of? You should remember some of this from talking about organic compounds.
DNA is a nucleic acid, and it's made up of the repeating monomers nucleotides.
So remember, a monomer is just the building block or the subunit that helps to compose that nucleic acid.
So the nucleotide structure has a phosphate group, which is this circle, a 5-carbon sugar, which is ribose, and remember if it ends in ose, it's the sugar. And if you look up, this is a pentagon shape. It has 1, 2, 3, 4, 5 points. At each one of these points, you have a carbon. That's why it's called a 5-carbon sugar. And then you have your nitrogen base. You're going to see later your nitrogen base looks a little different, but in the basic structural diagrams, you'll see it look pretty simple. You have 4 nitrogen bases for DNA. The first one is adenine, which is represented by an A. Then you have thymine, which is represented by a T. Guanine, which is represented by a G. And cytosine, which is represented by a C. So, where and why? DNA is located in the nucleus. Some can be found in the mitochondria and the chloroplast. So, what I would start here is endosymbiotic theory.
We just finished talking about evolution. We talked about how the cells on early Earth evolved from anaerobic prokaryotes to aerobic prokaryotes. Remember, if it's a prokaryote, it does not have a nucleus. If it's anaerobic, it does not need the presence of oxygen. If it's aerobic, it does need the presence of oxygen. And lastly, this evolved into your eukaryotes, which are also aerobic, which we are an aerobic organism. We're an example of a eukaryote because we're an example of an animal cell. So remember, when you had your anaerobic prokaryotes, they had mitochondria. You had your aerobic prokaryotes, they had chloroplasts. So through that evolution of those organisms, the genetic information that was embedded in each was carried from the mitochondria to the chloroplast and then into the nucleus, because the eukaryotes now are what currently hold our genetic information in our nucleus.
So, DNA controls the production or synthesis of proteins.
And you need to remember that your ribosomes are what actually produce proteins, so your DNA gives the blueprints or the instructions for producing those proteins.
How is DNA related to genetics? Well, you have your chromosome, which you all will inherit, or an organism inherits. Then you have the gene. So a chromosome is going to make up a gene. The gene is then going to compose or be embedded into the segment of DNA. That DNA is then going to create a protein. And then that protein is going to be used to express a trait.
So your eye color, your hair color, your shade of your skin, those are all traits.
So more about the structure of DNA. DNA is made of 2 strands of nucleotides. This is why it's called a double helix.
So in your brain pop video, you may remember it also told you that a DNA molecule looks like a twisted ladder.
The base pair rule, this is important. You need to remember that A, which is adenine, is always going to pair with T, which is thymine. And guanine is always going to pair with cytosine. So the same thing could be said in reverse. So C will pair with G and T will pair with A for DNA. They are held together by a weak hydrogen bond or weak hydrogen bonds. So what it asked you to do in your notes was to label the parts of the nucleotide on the picture. And you need to label it with the ribose, which is your sugar, your phosphate group, your nitrogen base, and your hydrogen bonds. So you already have this picture for you. You just need to label. So your pentagon shape is your ribose sugar. Your phosphate groups are these little circles. Your nitrogen bases are your C, your G, your T. So you can just pick one and label nitrogen base. And then this little white space that was filled in the middle, I tried to put little purple lines so you could follow with your paper. So those purple lines would be where your hydrogen bonds lie. Remember, that's what's going to hold
together your DNA structure. So replication, DNA replication. To replicate something just means to make a copy of it. So DNA replicates when, before a cell divides. What is the goal of DNA replication? To create an exact copy of the DNA molecule. And this process has a few steps. So the first step, the double helix unzips and the hydrogen bonds are broken. So remember, the hydrogen bonds are weak, so they're going to be easily broken. The complementary nitrogen bases in the nucleus bond with the unzipped strands. So those complementary nitrogen bases that it's talking about, that's when it's referring to your base pair rule, that A matches with T, T with A, G with C, or C with G. So the complementary nitrogen bases in the nucleus bond with the unzipped strands. So then step 3, 2 new molecules are produced. This is a semi-conservative process. This means that each molecule has one strand from the old molecule and one strand from the new molecule. So on this last slide, we have a picture that you don't have to draw because it's already on your notes, but just to give you an understanding of what semi-conservative means. So this is our original DNA strand right here. So then it's going to unzip, or our hydrogen bonds are going to be broken. So that's why you see your TG over here and your ACC over there. And then step 2 says that your complementary bases are going to line up with the unzipped strands. So that's why you see here with that G, you have a C, C, a G, A with a T. Same thing over here. So what's going to happen, they're going to bond together. From that bond, you're going to produce 2 new strands. So here is your new strand right here coming in. Here's your new strand over here. Together, those new strands are going to bond with old strands to give you 2 new molecules, a total of 4 strands. So 1 new strand and 1 old strand here. That's 1 new molecule, 1 old strand, and 1 new strand over here. That's 2 more strands, a total of 4, and then 1 additional molecule. So you can repeat the same process with these 2. So these 2 molecules would unzip because the hydrogen bonds would break. Then the complementary bases would line up with each of the old strands. And then you're going to have an old strand, new strand that will bond together to give you a total of 4 new molecules and 8 total strands. So you practice some of this on your paper, and good luck.